Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear student, we are studying now a chapter that is of class 12th, Principles of Inheritance and Variation. Dear students, in some of the previous lecture, we have completed with the mentalism, and in that we have discussed mainly monohybrid and dihybrid cross and the laws associated with that. Some of the questions we have dealt. Now, after this, today we are going to discuss some of the special crosses, some of the special crosses. And in the special crosses, we are going to understand back cross and reciprocal cross. First, we are going to deal with the back cross. Now, if you talk about a back cross, back means to the previous. Okay, simply it means. So it is defined as when an FN individual or heterozygote, FN individual will be always heterozygote. Okay, or a heterozygote of the next generation as well are crossed with any one of its parent or individual that are phenotypically and genotypically similar to the parent. It is generally called a back cross. So it is a backing, it is a crossing with the parents, the crossing with the parents which are phenotypically and genotypically similar. Okay. Now, in this back cross, we are going to discuss two type of cross. Okay. Then in that two cross, the kind of back crosses are outcross and test cross. Okay. Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid cross. Here we are going to discuss outcross and then we will discuss backcross. See, what is outcross? So outcross is crossing of F1 organism with the homozygous dominant parent. Okay, outcross is a crossing of F1 organism or heterozygote with the homozygous dominant parent. The first thing, one of the important thing is that it is not useful in genetics actually, but then also it was understood, it was defined, so we have to understand it. If we take an example, then in example, if we talk about a monohybrid outcross, then it is crossing of F1 organism with the homozygous tall. So F1 is tall, capital T small t, and it is crossing with the homozygous tall. So it is also tall, it is also tall. Here two gametes are going to be produced, capital T and small t. Here only one type of gamete is going to be produced, that is capital T. Now when capital T cross with the capital T, it is going to give rise a capital T tall. And if it is crosses with the this one, then it is going to form a capital T small t, that is all, that is tall. So here, if we talk about phenotypic ratio, then all are tall. So no ratio is obtained actually, 100% tall are there. Where genotypic ratio, if we talk, then it is capital T is to capital T small t, and that is 1:1. Okay, so this is the monohybrid outcross. So in monohybrid outcross, the first thing is that if we take a particular any heterozygote or any F1 organism and produce like this, you will get all tall. So actually, it is not having a no significance. Okay. Same way, after considering if here you will get a 1:1 ratio, but after studying it, if you discuss the dihybrid outcross, then dihybrid outcross. Now in dihybrid outcross, F1 is crossed with homozygous dominant parent. Now you know that F1 is capital R, capital Y, small y. And if it is crossing with the capital R, capital Y, which is a homozygous dominant parent, then here gametes are going to be capital R, capital Y, capital R, small r, capital Y, and small y. But here you are going to get only one type of gamete, that is capital R, capital Y. So if a cross is made between this, then you will get a capital R, capital R, capital Y, capital Y. Here you will get a particular result, that is capital R, capital Y, small y. Then you will get a result that is capital R, small r, capital Y. And next one is the small r, capital R, small r, capital Y, small y. Okay. So these four genotype are obtained in 1:1:1:1 ratio. So in the hybrid outcross, the genotypic ratio which is obtained, that is 1:1:1:1. Whereas if we talk about a phenotypic ratio, then phenotypic ratio is going to be all round and yellow sets are obtained in this. So this is the outcross, monohybrid and dihybrid. So sometimes you will get a question that what is outcross? Then you should be able to answer. Okay. But actually it has a no significance, because initially it was discovered and someone shows such significance, but later on it was discarded. So, and due to the discovery of this, another cross which was evolved, that is test cross, and it is of too much significance. In test cross, here also crossing of F1 occur with the recessive parent, that is a homozygous recessive parent. And in a typical test cross, an organism showing a dominant phenotype crossed with the recessive parent. In this test cross, what? Dominant phenotype is crossed with the recessive parent instead of self-fertilization. Now, why it is taking place? Because if we have a one plant which is a tall, by seeing it, we can't say that it is a homozygous tall or heterozygous tall. Okay, we can't say. So if we want to identify that this dominant trait containing parent or dominant trait containing plant or organism has which type of genotype, that can be identified by test cross. So it is useful in that way. Okay. So if we talk about it, as we have discussed, just test cross is a cross of F1 organism with homozygous recessive parent. Okay, so this is the thing. Now it is very useful. Okay. Now here it is used to find out the genotype of organism having a dominant form. Okay. So if we perform and understand it by some of the example, and we first carry out monohybrid test cross. And monohybrid test cross means it is a F1 is going to talk particularly cross with the homozygous recessive. So we take a case 1. In case 1, suppose the parent plant is homozygous tall, okay, F1, and it is crossed with the small t. then it is going to produce a capital T small t. Okay. So obviously it is giving rise to that all are tall plant. So if we make a cross of a tall plant, any tall plant with the dwarf plant, and if we get all are tall, then we have to understand that the particular tall plant has a homozygous dominant genotype. But if we take case 2, if that particular parent plant is heterozygous tall in F1 and it is made across with the small t, here you will get a two gamete, capital T, small t. Here you will get only one gamete, that is small t. When you will make a cross, you will get a capital T small t and you will get a small t small t. It is going to be tall phenotypically, it is going to be dwarf. So you will get both type of tall and dwarf in 1:1 ratio phenotypically, then that is a heterozygous tall. So if the result we get, that is tall and dwarf, by crossing a tall plant and dwarf plant, then we have to understand that the tall plant is not nothing but it is a heterozygous tall. Okay, in such way you can get a particular. Yes, so here phenotypic and genotypic both ratio are 1:1. Now after this we perform a dihybrid test cross. In dihybrid test cross, if we perform, then F1 is a capital R, small r, capital Y, small y. Okay, here also we can take particularly, here different possibilities are there. Okay, now I am taking just two cases. Case 1, if there is a capital R, capital R, capital Y parent and crossing with the small r, small y, you know that here capital R, capital Y gamete is going to be taken here, small r, small y gametes are there, and here offspring will be capital R, capital Y, small y, that is round and yellow. So if all round and yellow offsprings are obtained, then we have to understand that the taken F1 offspring or is nothing but what a particular or any particular parent is the capital R, capital Y, or dominant trait. Another case, if we take of a dominant trait containing plant, then that is if it is heterozygous for both character capital R, capital Y, small y, and crosses with the recessive parent, and here obviously four types of gamete are going to be produced, capital R, capital Y, okay, then capital R, small r, capital Y, and small r, small y, and here you will going to get a small r, small y. You will, if you make a cross, then you will get a capital R, capital Y, small y, then capital R, small y, small y, capital R, sorry, small r, capital Y, small y, and the small r, small y, small y. Here, if we take, it is a round yellow phenotype. Okay, then here the phenotype is round green. Here it is wrinkled yellow according to the law of dominance, and this is wrinkled green. So here, if we take a phenotypic ratio or genotypic ratio, the phenotypic ratio is 1:1:1:1. Okay, and same way genotypic ratio is obtained 1:1:1:1. So if this ratio is obtained, then obviously the dominant trait containing plant is heterozygote, whereas if the all are obtained in 100% probability of a round yellow, then the particular particular taken plant is homozygote. Okay, so this is the thing about the test cross. So test cross help us to identify the homozygosity or heterozygosity of the dominant trait containing parent plant. Either it is dominant or dominant homozygous or heterozygous, that can be identified by only doing a test cross and the results of test cross. Okay. Now, so that is test cross. So the progenies of such cross can easily be analyzed to predict the genotype of the test organism. The results of the typical cross where the violet color of the flower is dominant over the white color. If the F1 hybrid or in general a heterozygote is crossed with the parental type having a dominant trait in homozygous condition, no recessive individual are obtained. See, first it is the first case. When F1 hybrid is crossed with the parental type having a dominant trait in homozygous condition, no recessive individual going to be obtained in a progeny. So this is the cross which is given in NCERT. You can see this is the test cross. Alternative one is given where you can see here that the violet flower is crossed with the recessive homozygous recessive, which having the small p gametes and capital P capital T gametes, and then all are the heterozygous. All offsprings are purple color. Therefore, unknown flower was homozygous. No flower was homozygous. So this is, if this case is obtained that all are heterozygous and having a dominant trait, then obviously the parent is going to be homozygous dominant. But if the cross is made between heterozygous dominant and the recessive one, then half of the offspring will be of purple color and half of the offspring will be of white color in a 1:1 ratio. Means therefore unknown flower was heterozygote. Okay, so this unknown flower is of heterozygote, whereas this unknown flower is homozygous in this condition. Okay, so let's talk about some of the MCQs. Test cross means crossing F1 with dominant parent, recessive parent, both dominant and recessive. None of this. Actually, it is a crossing of F1 with the recessive parent. Okay, so that two become the correct answer. Test cross is a type of monohybrid cross, dihybrid cross, back cross, or none of this. We have studied this. It is a type of back cross. Okay, so third is going to be correct answer. Back cross means crossing of F1 with dominant parent, recessive parent, any one of its parent, another F1. So if you understand according to the genetics, what we have studied, the F1 individual or a heterozygote are crossed with the any one of the parent. So any one of the parent become going to be correct answer. That is the third option. Okay. Now after the back cross, one more type of special cross is remaining. That is reciprocal cross. Now if we talk about reciprocal cross, it is also known as reverse cross. Now what is done here? Here the same cross is made by changing the gender of the parent. Okay. So when a same cross is performed by reversing the parents it is known as a reciprocal cross. Now if we take an example, in first case, if we are taking a tall plant and if we are taking a dwarf plant, in first case we are taking a tall plant as a male and dwarf plant as a female. Means from the tall plant we are going to use a pollen grain or male gamete, and from the dwarf plant we are going to use the egg. They have a genotype capital T, and they have a genotype small t. Now their F1 offspring will be obtained, that is a capital T small t, that is tall, and when selfing is done, they give rise to the capital T is to capital T is to small t in 1 is to 2 is to 1 ratio, and the phenotypic ratio is 3 is to 1. You know that. Okay. Now, same cross is performed by a reversing a sex, then it is known as reciprocal cross. So here we are taking a tall plant and dwarf plant, but from here tall plant we are taking a female and dwarf plant is taken as a male. So gametes, male gamete are going to be used of dwarf plant, and the female or eggs, female egg is going to be used from the tall plant. Now if a cross is made between these two, then capital T small t tall is going to be F1, and if selfing is done, there is the result are capital capital T is to capital T small t is to small t small t. So genotypic ratio here is going to be 1 is to 2 is to 1, and same phenotypic ratio is going to be 3 is to 1. So there is a no change in result by reversing the sex. Okay, but such a way if a cross is made, then it is known as a reciprocal cross. Now the question is, what is the use of it? How it is useful? So actually it is known that this cross is done to find out the effect of parental sex on the inheritance of character. Okay, because see, okay, parental sex means if any particular trait is giving from a dwarf plant, okay, if it is inherited by a female plant, then it is going to come through the dwarf plant. But if it is any character is expressed by especially by female, not by male, then here it is going to come with the tall plant or tall genotype. Okay, so that is the thing. So if we talk about use, then this is crosses where this cross is done to find out the effect of parental sex on inheritance of character. Okay, so that is the thing. Okay, but result they are same in all reciprocal crosses. So that is the what a reciprocal cross, and these are the all type of special crosses. Okay, I hope you understood the uses of it and how they are performed. Thank you.